Hi everyone, welcome back. So now we're going to start talking about how we get proteins from the genes in DNA. So when we want to make a protein, we have to start with a gene, a coding region of a DNA molecule. Now the sequences of those nucleotides, something we often call bases, these are what contain the code for the sequence of amino acids that form that protein. Now, the sequence of those nucleotides is meaningful, and it has a clear start and a clear end. Now, DNA itself can have coding and non-coding regions, genes and non-genes. Now, these coding regions can be spaces between genes, what we call intergenic space. Not all DNA is made up exclusively of genes. And we can also sometimes have spaces within genes, where a single gene is broken up into coding and non-coding regions. So the coding regions here would be exons, and the non-coding regions would be introns. Importantly, if you see this situation in an organism, you're going to see that the gene always starts then with an exon, then you have an intron, then you have an exon, and so on and so on and so on, and it will always end with an exon. Importantly, this is not something we often see with bacteria. Bacteria don't have a lot of different kinds of introns. They have some, but most are not present here. This is mainly something we see with eukaryotes like us. So say that a cell wants to translate a gene into a protein, like insulin, for example. Now, the cell cannot use the DNA on its own as an instruction book as is. The DNA stays put wherever it is, whether it's in the nucleus in a eukaryote or it's in the nucleoid region pinned to the cell membrane in prokaryotes. It always stays there. Instead, a copy of that target gene is made, and then that copy can go anywhere. To accomplish this, the cell will transcribe the DNA as a copy, making a new molecule called messenger RNA, or mRNA. This is a single-stranded copy of a singular gene. Structurally, RNA is really similar to DNA. It's also made of nucleotides. It is also built on a sugar-phosphate backbone. But there are some important differences. For one, that sugar that makes the backbone is a little bit different. Ribose instead of deoxyribose. This is why RNA is called RNA and not DNA. It is also single-stranded, not double-stranded. There is not another side to this RNA molecule. It is just a copy of a single gene, so it's also only a copy of one strand of that molecule. And then there's also a nucleotide difference. Instead of RNA being ATCG, like DNA is, we have a substitute for T. RNA polymerase attaches to that promoter site. Then we move on to the next step, which is elongation. This is where RNA polymerase is going to be actively constructing that new strand of mRNA. It's going to grow longer and longer and longer as the enzyme moves across this template strand of DNA. Now, RNA polymerase always moves in the same direction. It always builds mRNA from 5' to 3' on the RNA strand. I want you to remember that. If we're always building from the 5' to the 3' on the RNA strand. So as RNA polymerase moves across the DNA strand, it is going to build the new RNA molecule from the 5' side to the 3' side, always adding complementary nucleotides to the 3' side of that molecule. The last step in the process is termination. So this is where RNA polymerase will reach the end of that sequence. There is a special region at the end called a terminator. And once the RNA polymerase reaches the terminator, transcription stops, and RNA polymerase is going to detach from that template strand of DNA. Now we have a new mRNA molecule that separates from that template strand, and it is able to go elsewhere to finish the next part of the process. That final part of the process where we are going to take our transcribed instructions to make mRNA and we are going to translate it into amino acid sequences to make the protein is called translation. Now, mRNA is an unstable molecule. It's not going to last all that long. It's going to slowly degrade, especially from the ends. Little bits are going to get broken off very easily because it's not as stable as DNA is. mRNA only exists for the time necessary to make a few copies of that special protein that it codes for. Now, mRNA is going to be shipped to another region of the cell to find something called a ribosome. A ribosome is that special enzyme whose sole purpose is building proteins from those mRNA instructions. It does that translation process. Now, a ribosome is also made of RNA. Technically an enzyme, but it's something special we call a ribozyme. It's an enzyme made not of proteins, but of RNA. So importantly, it still has all those familiar qualities of an enzyme. It is a facilitator of a reaction, and it is a catalyst, meaning that it facilitates that reaction without being used up in the process of that reaction. Now, a ribosome must read the mRNA and translate it into amino acids. I want you to think about the code of nucleotides in the mRNA as a language that must be translated, it must be decoded in order to make sense for you and for the ribosome. Every 3 nucleotides is what we call a codon. This is like a letter in the amino acid alphabet. And some codons are dedicated to what we think more like grammar and syntax, things like a start point and a stop point alert. We call those start codons and stop codons. Together, all these codons are going to communicate a series of amino acids that are going to build a specific protein. And there's some important things to note. I want you to take note of the position of nucleotides in the codon. This will be important later. So we have the first position being the first nucleotide in the codon, second one being the second, third being the third, obviously. I want you to also note that there will be some situations where multiple codons will code for the same amino acid. There will be some situations where they'll be represented by multiple codons. And this is because the genetic code, as we call it, is made up of many different codons, but there are a fewer number of possible amino acids. We only have 20 different kinds of amino acids that we can use to build a protein from. And then we have start and stop codon signals. There is going to be some overlap here. Now, to form a proper enzyme substrate complex, we need a couple things. 3 materials are needed. We need that ribosome, the ribozyme, the actual enzyme that is doing this process. We need the mRNA molecule, the set of instructions to build that protein. And then we also need tRNAs with an amino acid in tow. Now, a tRNA is also called a transfer RNA. It is a small piece of RNA whose job it is to carry the specific amino acid that is needed next in the sequence to the ribosome to add to the sequence. Every tRNA carries a specific amino acid and has a specific anticodon on the other side of the molecule. Those anticodons are a complementary set of 3 nucleotides that bind to a specific codon on the mRNA molecule. So for example, if you add a CCA codon on the mRNA, that's going to bind to only a GGU anticodon that is present on the tRNA. This is how the cell makes sure that only the amino acid that is coded for in the codon is brought and attached to the sequence of amino acids when we are building a protein. The ribosome will facilitate that codon-anticodon binding, and then it will do the hard job of grabbing that amino acid and separating it from the tRNA and attaching it to the long chain that begins to develop. The ribosome is going to do that job of linking amino acids together in that chain. Steadily, slowly, we're going to be building that all-important protein polymer. Eventually, we are going to reach the stop codon on that mRNA, and the sequence of amino acids is going to be completed. Then we're going to get the splitting of that protein from that complex. It's going to leave and become its own thing. Only when the amino acid sequence is complete, when the protein has achieved its final sequence, does it begin to take on its final shape. Now remember, protein shape determines protein function. So it is necessary for that protein to start to interact with its environment and itself and form these complicated shapes as it twists and tangles on itself in that extremely predictable way. Only then will we get a functional protein at the end. Now, the language of RNA, the translation of RNA into protein, it's important to note that is not identical across life. There will be slight differences that exist in the genetic code from organism to organism, mainly big groups of organisms, like comparing bacteria to animals, to fungi, to protozoans, and so on. But the basics still remain consistent. We have codons that relate to specific amino acids, that relate to specific start and stop codons, and the sequences of those things translate into an amino acid sequence to make a protein. This is a system of converting information from one form to another, from one molecule into another form of molecule. We call this the central dogma of protein synthesis. It's this core model that exists in all living things that moves information from DNA to RNA to protein.